Good afternoon, everyone, and I hope that you all have been enjoying the Harmattan school so far. And thank you very much for the opportunity to share a bit of perspective with you on the basics of radiation biology and artificial intelligence. Now, these are a bit of complicated specialties, but I'll try to present them in a simplistic approach. You might be wondering, AI and biology, aren't they supposed to be contrasts? Well, they seem so, but today I'd like to share the connection between these fields, especially as they apply to medical physics and cancer research. So please join me in the lecture. So there are two bases for this presentation. One is to understand the biological effect of radiation on the human body, looking at the indirect action and the direct action mechanisms, and also to understand how cancer cells and normal tissues respond to radiation, and to also sort of look at how the DNA is a principal target for damage in the human body. And of course, in the next section, we're going to look at artificial intelligence. And why AI? Because I'd like you to look at AI from the human body perspective. The human body is an intrinsically complex network of data sets that sort of creates functions for our existence. Data sets like the cells, the enzymes, the microbiomes, the molecules, and etc. And all of these are really working together for our good. So the pattern in the human body is compared to a neural network on which the principles of deep learning is actually based. And understanding all of this would sort of give us, a clearer perspective on how AI can be fused into radiobiological research and ultimately for medical research. So now, let me take you down memory lane. I believe that you've actually seen this over time during the course of this presentation. So I'm not going to sort of, be repetitive, but of course, repetition is very key to persuasion, and understanding history is very critical to securing the future. So let's sort of look at how the field of radiation biology began. So in 1895, William Courant Rothstein made a significant discovery by discovering X-ray, which you probably know already. And of course, in 1896, Becquerel and Marie Curie discovered the phenomenon called radioactivity. But in 1902, a tragedy happened. What happened? The first recorded biological effect of radiation was reported, and this was actually because Becquerel sort of left a radium container in his pocket. And as you can actually see from the slide, this is actually Marie Curie, the husband of Mary, who actually repeated the same experiment on himself to actually see if Becquerel's experiment was true. And he placed a radium source on his hand and later recorded a skin ulceration. This was interesting, but also intriguing, because even though that scientists have discovered that radiation could, help us understand the inner workings of our anatomy, on the other hand, it could also cause biological damage. And this discovery began the field of radiation biology. So in 1912, several societal and regulatory bodies were formed to prevent patients, personnel, publics from the harmful effect of radiation. And I must actually tell you that we have indeed come a long way in the science of radiation protection. So there are several professions that are actually working together to ensure that cancer research improves. And you can take a deeper look into this in your PDF document. I call it the Wheel of Progress. Now, you've probably seen this over time during the course of the Harmattan school. So what does a medical physicist do? We definitely use the principles of the electromagnetic spectrum chart to solve varieties of problems. So let's go into the lecture, radiation biology. So in this paper, we sort of looked at, the progress of radiation biology research for over a century, from 1930 to 2020. And we actually reported that the field of radiation biology has seen an impressive progress in terms of research output. But however, in the year 2000, we began to actually see a steady rise in research progress, and a spike was actually noticed in 2010. And we believe that this spike was actually due to the influence of artificial intelligence applications in medicine. So here I came, guys, and there was a boom, and I can actually tell you that there is still a lot of work to be done in this regard. But let me clarify to you that even though in 2010, AI came and there was a boom, but there has been a long use of AI in medical research, dating back to the 1950s. And however, different applications actually started in 2000, due to the influence of, deep learning applications, which the principles of neural network is based. So let's go into radiation biology. So I believe that there are three roles by which radiation biology actually plays in cancer research. One is, understanding the tumor response to radiation and normal tissues, the mechanisms that underlie this response. And second is understanding biological radiotherapy. This is basically using biological factors, to further improve radiotherapy treatment. And thirdly is to sort of use biological factors to improve treatment protocols, especially in the areas of drug discovery. So the image you can actually see shows the timescale of events of radiation exposure on the biological systems. So the biological phase, I believe, is actually the most critical of all phases. This is not to downplay the other phases, but you can actually see something very amazing in this diagram, that the physical and the chemical phase only occurs within fractions of picos or microseconds, while the biological phase, occurs for an entire human lifespan. And this sort of, informs us about the most important reason to sort of study radiation biology at the microscopic levels. So let's break it down into two, radiation and biology. But first, let's start with biology. So you probably remember what biology is, right? Biology is the study of what? Life, right? You got it. So it's sort of understanding, the physical processes, the chemical structures, the developmental and evolutionary processes of living organisms. And from high school biology, you probably remember what is called the systems, the organs, the tissues, the cell structure, right? Great. So what really is the body made up of? Because I believe that if you understand this, it will sort of broaden, our perspective and understanding on radiation interaction with the human body. So the human body is made up of invisible organs and the invisible microbiome, the things that we cannot see. And a combination of these two leads to what we call the normal functioning human body. And statistically, we've seen that there are an average of 100 trillion atoms per cell, amazing. An average of 100 cells per body, super amazing. 100 million microbes and 9 million genes. So it actually tells you that locked down on the inside of you are trillions and zillions and billions of atoms and molecules and proteins and enzymes. And understanding this would sort of help us to, have a clear perspective on how radiation interacts with the human body. So let's look at the brief summary of how radiation interacts with the human body. During the previous lecture, amazing speakers have actually done a lot of justice to this. So I will just sort of flip over. We know radiation is divided into several types: electromagnetic radiation, particulate radiation, and recently, we have gravitational waves, acoustic radiation, and, they have different classifications. But basically, when radiation interacts with the human body, it could directly ionize or indirectly ionize. What is the difference? And I'm going to spend much time on this because this is the basis for this course for radiation biology. So in the directly ionizing interaction, the secondary electron, the photon that is, interacts first with the DNA and therefore induce a biological damage. Please take note of the word in red, interacts first with the DNA. But what is the difference in the indirect interaction? The secondary electron, the photon that is, interacts first with the water molecules. Why? Because the human body, is made up of 70% of water. There is an old chemistry joke that if you like me, if you like water, you already like 70% of me, because the human body is made up of 70% of water. So in the indirect interaction, the secondary electron first interact with water molecules. Water molecules produces free radicals, that the free radicals result in damage to the DNA. And when radiation interacts with the human body, we could have two types. It could interact by ionizing interactions, or it could interact by non-ionizing interactions. I'm going to explain that briefly. So in the ionizing interactions, there are sort of electromagnetic radiation, that have sufficient energy to ionize atoms or molecules by detaching electrons from them. And we have three types: alpha particles, beta particles, gamma rays, which you probably know from the previous speakers. And of course, we have the non-ionizing radiation. Currently, some of you are actually receiving non-ionizing radiation as we're speaking. You're looking at me from your laptops or from your phone and thereabout. And in this interaction process, the molecules really does not sort of have sufficient energy to ionize. The energy that is produced is only sufficient for excitation process only. And what is excitation? The increase or the move, of electrons from a low energy state to an high energy state. And I must actually say this because it's very important. We often gloss over this when we're talking about non-ionizing radiation. Why there are different biological effects of radiation that have been reported from non-ionizing radiation, I believe that the effects are non-deleterious. Now, my colleagues in health physics might raise an eyebrow, but however, the International Agency for Research on Cancer recently stated that there could be some risks from non-ionizing radiation to humans, and it's very important to state this. So coincidentally, I believe that the science of physics, chemistry, and biology agrees that radiation damage to the human body begins at the microscopic levels, the atomic levels, and at the molecular levels. And speakers have actually done justice to the physics point of view. So I'll take you straight down to the biological point of view. I already explained this. So when radiation interacts, it could first, create a direct target, to the DNA, and of course induce mutations, and of course there could be different biological responses. Or in the second phase, it could sort of, ionize water molecules. Water molecules could create, hydrogen peroxide or free radicals that induce mutation, and of course we have a biological response. But something is happening here, like I already mentioned in the previous slides. Now the physical steps that actually leads to the energy deposition and free radical formation often occur within nanoseconds, right? While the biologic expression of the physical damage or the biological response occurs from minutes, to decades, and this can be deleterious, resulting in carcinogenesis. I believe this is the reason why we must pay attention to biology, even as physicists. So the DNA is the principal target for biological damage in the human body, and I'm going to be focusing on this. So what is the DNA? The DNA, as you probably know, is deoxyribonucleic acid. It's a large molecule with double helix structure held together by the adenine-guanine bases, the adenine-thymine base, and the guanine-cytosine base. You've probably heard of this ATGC. Okay, so let's look at a structural view. So in this first diagram, we have a normal DNA helix, right, showing the hydrogen bases. And we also have, in the second diagram, we have a single strand break, which is oftentimes repairable. But where is the break? Can you see the break? We can actually see a loss of sequence here. But in this case, it is repairable because it can actually copy the sequence of this and get itself repaired. In figure C, we can actually see a double strand breaks. Where are the breaks? We can see break 1 here, break 2 here, but the break is repairable because they do not occur at opposite ends. So this could copy the sequence of this and get itself repaired, and this also repeat the same process. But in the situation when the DNA DSB damage leads to chromatin breakage, and oftentimes not repairable, is when there is the breakage at opposite ends. And of course, this further shows us why the DNA is the principal target for biological damage from the effect of radiation. So when mammalian cells receive radiation, it could result in 3 types of damage. One is the lethal damage, which is not reversible, not repairable, and oftentimes it's a cell death. The most lower form of damage is the sublethal damage. Sometimes it is repairable if it does not receive additional dose of radiation, and oftentimes repairable in hours, but ultimately also leads to cell death. The last is the potentially lethal damage, sometimes modifiable by the radiation cell conditions, and oftentimes also leads to cell death. So what is cell death? I believe that understanding this is very critical. So cell death is the permanent loss of clonogenic capacity or reproductive integrity. What does that mean? When cancer cells, receive radiation, or before they receive radiation, cancer cells by mutation process grow uncontrollably. They divide and they mutate, and they go rogue, and they grow and grow and grow to the form of tumor, to the form of growth and all of that. But when cancer cells receive radiation and they do not have the ability to reproduce and proliferate and grow, then there is a loss of clonogenic capacity. But what are the differences between a normal cell and a cancer cell? Please look at the image. So in the normal cell, we have a very fine cell membrane. We have a very fine cytoplasm. From some of you that still remember your biology, the cytoplasm, that is the cell environment, is still very much, bigger. We have a very fine chromatin. But in the cancerous cell, what can you actually notice? The cell membranes are very irregular. Then the cytoplasm begins to decrease. Why? Because the DNA now, has been damaged, and of course it gets multinucleated, meaning that it has multiple nucleus production. And of course, this is one of the reasons why cancer cells get mutated, which is actually due to the presence of apoptosis. And apoptosis really is one of the basic reasons for cell death. It's called the programmed form of cell death. And it's like the process of the cell trying to eliminate an unwanted form of cells. And apoptosis really maintains the balance of cells in the human body. And I must say that it is extremely important for immune system regulation. So let's look at some cases. So cells cultured in a laboratory, we have metastatic breast cancer cell, which oftentimes develops from the breast tissue, as you can actually see. So we have 3 sets of dishes, dish 1, dish 2, and dish 3. In dish 1, the cells were not exposed to radiation. These are cancerous cells. In dish 2, they were exposed to 2 gray of radiation, and in dish C, they were exposed to 5 gray of radiation. So what is the difference? As the radiation increases, by the way, in the 0 gray dish, we can actually see that the cells are highly dense. We can actually see a lot of clustering going on here, right? They are highly populated. But when they receive 2 gray of radiation, we can actually see cell death. Look at these species. So the cells begin to die, the morphology begins to change, the phenotypic expression begins to change. And of course, when we increase the radiation, what can you actually see? A more increased cell death. This, of course, shows us that radiation has effect, on cancer cells, and we really want the cancer cells to die. That is literally what we do as physicists. So when cells are irradiated, the outcome could result in 9 different outcomes, which I'm going to explain in a brief. So in the first process is that there is no effect. Okay, in the second process, there is a delayed division. This is when the cell is delayed from going through division. And the third process is the apoptosis, which I explained. This is the reproductive failure, which is the first outcome, which I already explained. Remember the loss of clonogenic capacity. Yes, you remembered. Okay, so in the next phase, we have what we call the genomic instability. This is when there's a delayed form of the reproductive failure. The cell is unstable genetically. And of course, we have the mutation. Yeah, in this process, the cell receives radiation, but now the cell does not die. It has now gone rogue, and of course creating mutated genes. And in the next phase, we have transformation. This is literally when the mutation leads to transform phenotypic expression. And of course, we also have the bystander effect. This is when the irradiated cell can now communicate to the non-irradiated cell and induce damage. This is a very good area of research. And of course, we have the adaptive responses. So either ways, any cell that is irradiated will definitely receive this outcome. And one of the most forms of adaptive responses, when the cell sort of gets, adaptive to radiation, it becomes resistant to radiation, is the brain cancer cell. Also, maybe one of them is glioblastoma, and which I'm going to show in the case 2. So this is a brain cancer cell, specifically the name of glioblastoma. Glioblastoma multiforme literally is one of the most aggressive form of brain cancer, which we're currently researching, and it actually occurs in the frontal and temporal lobe of the brain. So we have 3 dishes as well, dish A, B, and C. We can see something interesting going on here, guys. Please pay close attention. In the 0 gray dish, we can actually see that the cells have a very distinct spreading, membrane. You can actually see the cells, are very, very distinct. But when they were exposed to 4 gray, first we can actually see a decrease in the population. We can actually see, some cell deaths. But when they were increased to 8 gray of radiation dose, what can we see? We can actually see continuous cell death, actually occurring. And of course, this sort of shows us, some of the effect of radiation, on cancerous cells. And of course, a paper published in a laboratory in 2019 shows us the radiation effect on cancer cell viability. If you look at all of these cancer cells, from squamous carcinoma to 4T1 to pancreatic cancer cells to metastatic breast cancer, we can actually see that compared to the unirradiated groups, there is a decreased cell number when the cells were irradiated. And of course, this further proves what I've been trying to say. So let me see whether you're following me. Let me give you a quiz. So cell A was irradiated with a dose of 4 gray, while cell B was not irradiated. Which of the cells do you think would result in a higher damage to the DNA? Please think for a minute. You might receive a gift. Just kidding. Okay, so hold your answer in your mind and let's look at the answer. But before you give me the answer, let's look at another case. Remember I told you that the DNA is the what? The principal target for biological damage in the human body. So let's look at the radiation effect on the DNA damage. So the picture you're seeing is an immunofluorescence experiment. Immunofluorescence, you've probably had a fluorescence from, I think, part 3 physics. It's the excitation of light from substances, from chemical substances, from biological substances. So this is an immunofluorescence experiment that was carried out also in a laboratory, and these cells are pancreatic cancer cells. They are cancer cells derived from the pancreas. So we have two sets. One was exposed to 4 gray of radiation, the other was not exposed. So, but by the way, they were actually stained, and with some markers to help us see what is going on. And it's after this staining that we can actually expose them to different wavelength of light to see the interaction going on. But in the blue section, in simple language, in the blue section, we can actually see the staining of the nucleus. So we can see the nucleus of the cell. In the red section, we can actually see the cytoskeleton. This is the cellular environment. And in the green section, we can see what we call the DNA damage. But what can you actually notice? In the blue section, not much of a difference between the 0 gray and the 4 gray. Same thing in the red section. But in the green section, something is going on here. In the 0 gray, we can actually see that there is little damage to DNA, right? But in the 4 gray, there is an increased damage to the DNA. And of course, this further proves, the radiation effect, on DNA damage. So let's go back to the quiz. What is the answer? Cell A, right? So please clap for yourself because of if you got it right. And of course, if you did not, also, give yourself a round of applause. So cells that are irradiated with an higher dose of radiation, will definitely result in damage to the DNA. So now in the next slide, it's a bit of discomforting. I apologize in advance, but to sort of show you the radiation effects on, the skin, radiation-induced effect on the skin. And these are images acquired by permission from the Hiroshima and Nagasaki artifact of museums, showing radiation exposure from the fallout of the 1945 bomb blast that happened in this city. We can actually see in this diagram of this little boy of radiation exposure effect, on his hair, causing epilation. Epilation means the loss of the skin. In this second image, you can actually see a dry discoloration causing deep skin necrosis. This is a soldier, a Japanese soldier. We can actually see skin reddening or erythema. Remember the Bequere bone, nice. And also in this woman, we can actually see keloids. We can actually see, cataract formation from high dose of radiation resulting in radiation retinopathy. This is severe thermal effects. Now, the reason why I'm showing this picture is that some of you might have been excited about how radiation can be used to cure cancer. Yes, it is true, but on the other hand, radiation can be very dangerous. And the question we must ask ourselves is that how far are humans willing to go in the applications of radiation? But of course, we must always stay on the positive side of the use of radiation. So I spoke about cell death, but can the cell repair itself? Definitely the cell can also repair itself by some mechanisms. I'm running out of time, so I'm going to switch over to some of the key points. So if you've forgotten everything I've said on the radiation biology lecture, please do not forget this important point. One is that radiation decreases cell viability and it changes the phenotypic expression, like you can see from some of the cases I showed you. Number two is that the DNA is the principal target for what? Biological damage, right? And number three, there are computer simulation programs that are definitely available for understanding, radiation biology interaction. You can always go on YouTube, to see how these programs could be downloaded, and of course you could also study them. And of course, number four is that understanding the molecular pathways behind cell response to radiation damage is very essential. I know this is biology, but of course too, it is very, very essential. And before we go into the artificial intelligence section, I'm going to show you a quote from Nobel laureate Francis Crichton. Is an American biologist who won the Nobel Prize in Physics and Physiology and Medicine in 1966. He says, and I quote, "Tumors destroy man in a unique and appalling way, as flesh of his own flesh, which has somehow been rendered proliferative, rampant, predatory, and ungovernable. They are the most concrete and formidable of human maladies. Yet, despite more than 70 years of experimental study, they remain the least understood." This statement was made 55 years ago. And I can tell you that even though that we have seen an impressive progress in cancer research, they still remain the least understood. But thankfully, to artificial intelligence, we have seen how cancer cells can be further understood. So let's go into the artificial intelligence section. So I'd like to show you what we call the hallmarks of cancer. These are actually what makes cancer cells what they really are. And of course, about 20 years ago, if I'm right, scientists by the name of Weinberg and Hanna sort of, introduced to us what they call the hallmarks of cancer. These are the characteristics of cancer, and for the purpose of the AI section, I'll be focusing on cancer invasion and metastasis. So now, remember I told you in the beginning that the human body, consists of several trillions of molecules, of atoms of cell. Sure, but the question I'd like you to think and ask is that how can we investigate the biological interactions at the cellular molecular levels? Mathematical models, machine learning algorithms, computational biologists, and of course, AI. So why AI? My colleague did a very insightful study in just recently, in the recent lecture on the applications of artificial intelligence in medicine. So I won't bother to reinvent the wheel, pardon me. But why AI literally? So why AI? I believe that artificial intelligence has a significant role to play in medicine. And of course, I also think that artificial intelligence algorithms can actually be used for analyzing extensive medical data sets, ultimately for the purpose of prediction and of course, detection of diseases. So what is AI? AI stands for artificial intelligence, and it's really truly a form of intelligence. But this intelligence is to sort of simulate or replicate, human intelligence in machines. And its purpose is to, develop or enable computer programs to be able to perform high intellectual tasks. And machine learning is a subset of AI, which definitely, uses algorithms that can learn without the assistance of human, and deep learning definitely uses neural networks. So let's see how the applications are useful in cancer invasion and metastasis. Now, remember I spoke about the hallmarks of cancer. Do you still remember? Sure. So let's go into defining what invasion and metastasis is. So cancer invasion and metastasis. So to sort of explain it in a local language, from the Nigerian perspective, invasion is like a marketer showing up in your apartment or in your room, or maybe in your house early in the morning on the Friday morning, knocking on your door and saying, please come and buy this thing from me. That is invasion of privacy. And what is metastasis? Metastasis is like MTN, the popular network everywhere you go. Even if you go to, some remote villages, you will still find MTN marks, where you do not expect to see civilization, you will definitely find an MTN mark. That is a case of metastasis. But to bring it into a scientific term, invasion refers to intrusion. So the cancer cells, when they originate from the primary point, which is called the original site or the primary tumor, they can have the ability to invade the neighboring sites. And of course, they travel, and of course, they metastasize into the metastatic site, also leading to the formation of tumor. So how can AI be applied? Definitely, into understanding the invasion and the metastatic process. I think there are 3 steps. One is understanding the roots of the metastatic process. Secondly is understanding the drivers of the metastatic process. And third, and most importantly, is understanding the destination of the metastatic process. You can actually see that the cancer cells, which are in red, are nicely fitted into this red line. And this red line is known as the pre-metastatic niche. So imagine that we have the ability to build an artificial pre-metastatic niche using tissue engineering, and we can load this artificial pre-metastatic niche with different drugs, with different tumor control factors, and we can have the ability to capture the cancer cells as they arrive this destination. And that is one of the reasons why I said understanding the roots, the drivers, and the destination is very, very important. This is a more complicated design of what the pre-metastatic niche looks like, and I'm going to just flip over that. So how can AI be applied to understand or combat cancer metastasis? 3 steps. Number 1 is in the case of metastatic detection. Number 2 is understanding how AI can be used to analyze pathological images, and of course in this last phase, which is by using engineered payloads to target cancer sites. And of course, there are several papers. Recently, one was published in 2020 looking at how AI was actually used in the diagnosis of bone scintigraphy. So let's take a diver deep. Now, I often tell my physics colleagues that how best can we understand, or how best can we cure cancer if we do not understand the biology of cancer? Yes, definitely. I am not undermining the role of physics. In fact, I have a physics background, but because of my multidisciplinary training, I tend to look at the cancer cell from a biological point of view. When physicists speak, they talk about, the photoelectric interaction, the Compton's effect, the Rayleigh scattering, the direct ionization interaction, and all of that and all of that. But oftentimes, physicists neglect the tumor characteristics of the tumor itself. And understanding the tumor characteristics, could sort of give us an inclination to use them as factors for driving deep learning applications. So these are some factors, tumor factors, that I think that could be very useful. So let's get into the section deeper in the few minutes that I have. So in case one, just like I did for the radiation biology section, in case one, I think that not that I think, it's scientifically proven that AI can be very useful in metastatic detection. So let's understand what metastatic detection is, specifically in lung cancer. So we have a case of a lung ear. We have the trachea bifurcating into the primary bronchi. We also have another bronchi here on the left, and of course we have the bronchial stem. So what happens here in the stage 1 cancer, the lung tumor can be localized, in a very small dimension, and of course the disease, has not metastasized outside of the lung. It is still very much localized in the lung. At this point, relatively treatable, definitely. And we also have what we call the stage 2 lung cancer. In this process, the cancer begins to migrate upwards, begins to develop inside the bronchial stem. At this point, yeah, relatively treatable. But in the stage 3, it becomes very difficult because now the cancer cells has begun to invade the neighboring environment, like I told you. But in stage 4, the cancer cells now has left the entire lung, travels through the brain to the liver, and of course metastasize in the brain. So how can AI be applied? Please think for a moment. So assuming that somebody has a stage 1 lung cancer, the cancer cell can be treatable in stage 1. But if we have an AI program that can definitely tell us that this is a potentially metastatic cancer that could be found later in the brain, the AI program would have alerted us just when this person has a stage 1 cancer, and would definitely help clinicians and oncologists to develop better treatment protocols. So let's look at some of the applications in the few seconds that I have. Please pardon me. So this paper published by some of our colleagues in South Korea in 2020 look at how exosomes can actually be used to sort of play important roles in cancer stage, early cancer stage, diagnosis prediction using a deep learning model called the principal component analysis. So the overview summary of this figure is using biological factors like exosomes, the data we actually acquire from the exosomes using Raman spectroscopy. And of course, the data we actually matched with patients, that either have a lung cancer or with patients that don't have lung cancer. And of course, the models were trained using the principal component analysis, and of course, using the Mahalanobis distance to understand how the correlation could be. So if there is, a shorter distance between the lung cancer patient's exosome data and of course the cancer cell exosome data, then of course it could mean that the stage is, at a more advanced stage. But of course, this is one way of using AI in predicting lung cancer. Another case is the pathological image analysis. I actually think that there is an intersection between AI and cancer pathology, and that is called the digital pathology. This is basically using machine learning algorithms to understand or create or reconstruct images. And these are some of the nomenclatures that are used. But let me go to the application in this slide. So this slide basically shows, an automated gleaning grading system of prostate cancer. So this diagram shows, prostate cancer tissues that are actually being graded by cancer pathologists. And this section actually shows, the AI grading system. And we can actually see that the AI sort of gives a perfect Gleason score, after the training process. And this is one way in which, AI can actually be used in the application of pathological image. And of course, in cancer registration, definitely applicable. AI sort of shortens the time in the way you take, in the way that cancer image registration is done. My apologies for rushing, but in the brief summary, I like to say that AI has had a long way in cancer research. And we still have a long way to go. Definitely we do. But of course, I think there are giant strides that have been recorded, especially in the areas of tumor imaging, cancer screening and diagnosis. For my example, when I'm in the wet lab studying cancer cells from a microscopic point of view, I can only make judgments as far as my visual eye can see. But when AI is applied to the images that we look at, of course, then we can have much more data to make for prediction and diagnosis. So let me give an overview, a summary of everything that we have just learned. Number one is that radiation is a double-edged sword. It can cure and damage at the same time. Number two, medical physics is a rapidly expanding field. Remember, I showed you with the wheel of progress. So please, you can always join us after you finish your first degree. And secondly, or thirdly, pardon me, is that making a novel contribution in cancer research requires a strong multidisciplinary collaboration. So some of you, are studying physics electronics. Some of you are studying physics engineering. Some of you are actually studying physics education or thereabouts. So there is a significant multidisciplinary collaboration that is needed. So your chemistry knowledge, your computing knowledge, your machine learning knowledge, all of those knowledge are actually needed to help us, fight, cancer, make a contribution. And the last, which is the most important, is that you are not too young to make an impact. I understand that most of you listening are probably in your fourth or third year, and I also acknowledge your senior colleagues listening, but you are definitely not too young to make an impact in the field. And just like I did by showing you a quote, I'm going to show you a last quote. This is a paraphrase, and it's an amazing quote. It says that AI will not replace medical physicists or any specialty of physics, especially this is a concern to some of our senior colleagues. But the truth is that medical physicists using AI will definitely replace those that are not using it, because AI is the future. So ladies and gentlemen, thank you very much for the opportunity to have this short conversation with you. I hope that you've been able to learn a couple of things, and please feel free to let me know if you have questions. Thank you very much again.